Thank you for that kind introduction, and thank you DDL committee for the award. It is an honor and privilege to be receiving the 2021's Emerging Scientist Award. Good morning, everyone. Today, I have the privilege to present my career's work on developing advanced integrated respiratory models that could aid the rapid translation of inhaled therapies to clinic. First, for some background, chronic respiratory conditions affect more than 550 million people worldwide, with respiratory infection and COPD named the third and fifth leading cause of death, respectively. Inhaled therapies have been the cornerstone in the treatment of patients living with these chronic respiratory diseases. The benefit of these therapies have long been recognized for their ability to deliver high drug concentration locally to the respiratory system, leading to improved efficacy and reduced side effects. While there have been significant progress within the field, only a handful of new inhaled products have been successfully progressed to market over the years. In fact, only 27 products have been approved in the past 5 years, in comparison to oral and injectable formulations that have 10 times more products approved in the same period. This is due to the complexity that is inherent in the development of inhaled therapies, where its efficacy is influenced by multiple interacting factors between the drug, formulation, device, lung physiology, and patient-related factors. The respiratory tract is a complex organ that is heterogeneous with multiple branchings and very different to other organs in terms of how we treat it, whereby the therapies are heterogeneously distributed across the airways once administered. The mechanisms of how the particles deposit on the different regions is very different, and so is the particle size that reaches that particular region. Generally, for the upper respiratory tract, such as the nasal, trachea, and bronchi, the larger particles deposit via inertial impaction, and in the lower regions, the smaller particles deposit via sedimentation and diffusion. Generally, characterization of respiratory aerosols is performed in 3 parts. Firstly, physical-chemical characterization of the formulation to evaluate drug dissolution, release, size, shape, charge, and thermal properties in response to temperature and humidity. Second is the aerosol performance and deposition, where we evaluate how the formulation and device performs together and see where it deposits in the respiratory tract. And finally, biological characterization to test the efficacy and toxicity of the formulation. Typically, these are some of the current methodologies employed to study the physical-chemical characteristics, specifically the dissolution rates of the formulation, which includes the rotating pedal, the flow-through cell, and France cell. However, these methodologies exist for testing of conventional solid dosage formulations, which are designed to mimic other organs such as the gastrointestinal tract, liver, brain, where large volume of solution is not representative of the lung physiology and with no regard to the effects of aerosol size distribution. Our normal in vitro testing for aerosol performance on inhaled therapeutics in the respiratory tract are also not so normal. Pharmacopoeia-approved impactors and impingers are used to segregate the aerosol particles into the different size fractions, providing an estimate of where the particle lands in the respiratory tract with outputs of mass median aerodynamic diameter and fine particle fraction. Yes, size is one very important consideration, but another important feature to consider is what happens after these aerosols land in the lung. Conventional studies are performed using drug solution or suspension that is significantly different to the in vivo real-life scenario of drug deposition in the respiratory tract, where aerosol particles are impacted onto the mucosa surface after aerosolization. While drug solution and suspensions may be representative of the environment in other organs, but deposition and the release of drug from microparticulate systems deposited in the respiratory system follows a series of processes due to their physiology. So upon impaction or deposition of the particles on the epithelium, the particles will encounter a limited volume of surface liquid or mucus on the epithelium that will subsequently be wetted, followed by dissolution of drug into the mucus layer, establishing a localized concentration gradient. This gradient will drive the permeability of compound across and into the cells, either through passive or active transport mechanisms, dictating the efficacy and toxicity of the drug unless cleared by mucous layer clearance or enzymatic degradation. Traditionally, model of the respiratory epithelium has been proposed and developed over the years, as they are the main barrier of drug absorption to study efficacy and toxicities. Previously, there have been two models of the lung epithelium that has been used for research, which are the liquid covered culture, also known as the wet model, LLI, which has media on the apical chamber. On the other hand, the air interface culture, also known as the dry model, ALI, has their media aspirated after 24 hours and cultured for a further 2 to 8 weeks depending on cell type to represent the lung environment. The dry model has been shown to generate a model more morphologically representative of the airway epithelium than cells cultured in liquid in terms of ultrastructure, secretory component, and electrical resistance. Here are some of the differences found. Studies have shown that the liquid-covered culture displayed short microvilli and clear cell-to-cell boundaries compared to the air interface culture where there is a heterogeneous population of cells with extended cilia and mucus production. Also, the air interface culture produces a thicker cell layer with apparent pseudostratification. And finally, the tight junctions in the air interface culture are less restrictive as seen with the more diffuse Z01 green dye, which is similar to epithelial in vivo. Now, existing models are confined to characterization of individual elements of aerosol characteristics, such as aerosol performance using cascade impactors and biological activity using cellular models as separate entities that does not interact with one another. These conventional methods are nowhere near close to being physiologically realistic, with existing studies consistently demonstrating an overestimation of the aerosol deposition in the human lungs, as well as its efficacy and toxicity on biological models. The failure to translate promising inhaled drugs to the clinic will continue to persist due to the absence of a predictive in vitro platform that could accurately mimic the physiological conditions and inhalation processes of the respiratory tract. My research program has directly addressed this problem with the development of advanced integrated respiratory models that capitalizes on the program of work that I've led over the past 10 years, which ties in pharmaceutical aerosols and advanced system biological techniques to understand the dynamic interactions between aerosols and the respiratory biological barriers. Firstly, I have established a series of physiologically relevant air interface biological models that closely mimic the lung. To this end, it is known that deposition of pulmonary drugs from aerosol devices are mainly in the airways where epithelium is the principal barrier to drug absorption. My team and I have established a number of epithelial air interface models that includes both commercial cell lines and primary cells of the different airway regions, including nasal, bronchial, and alveolar cells as listed here. These models have been optimized and characterized for inhaled pharmaceutical applications. They have shown to be fully differentiated epithelium with different cell populations and pseudostratification, expressing essential properties such as tight junctions, permeability, mucus, and protein expressions that are similar to in vivo lung physiology. More importantly, my team has also established protocols and characteristics such as passage number, seating density, and days in culture to ensure that the models are reproducible time and time again. To ensure physiological relevance for different respiratory conditions, my team have also been able to establish a number of co-culture models, including infection models with bacteria such as Pseudomonas or Staphylococcus, as well as biofilms. In addition, we have also developed several disease-specific models, including cystic fibrosis, lung cancer spheroids, emphysematous fibroblasts, and lymphatic cells to understand how inhaled therapies interact with the different cell types and diseases to provide a better mechanistic picture of what really happens in the airways. Subsequently, to mimic the in vivo deposition and evaluate how real aerosols impact on biological functions, the air interface models were integrated together with pharmacopoeia impactors as one system. In this section, I will highlight some of the integrated models that have been developed and provide an example of its application. The first of this model is a simplified twin-stage impinger that was used to deposit respirable particles of less than 6.4 microns onto an air interface model. The TSI was adapted to fit an air interface model at the second lower chamber where respirable aerosols can be deposited on the mucus layer of the epithelium. Subsequently, evaluation on drug transport, efficacy, and toxicity studies can be performed on these cells. What else can the model predict? In one of my previous studies, Two dry powder ciprofloxacin formulations were studied, which are spray-dried ciprofloxacin alone and controlled-release dry powder ciprofloxacin spray-dried with polyvinyl alcohol (PVA). The controlled-release formulation was supposed to be developed as a superior alternative to increase the residence time of antibiotic through the gradual and predictable release of ciprofloxacin and potentially reduce the burden of treatment. Both the formulations were found to have similar particle size, with the spray-dried ciprofloxacin alone having a corrugated surface, in contrast to the controlled-release formulation having a smoother surface with occasional cavity formation. Nevertheless, both the formulations are amorphous, as shown with the series of phase transition exothermic peaks on a differential scanning calorimetry that was performed to analyze the thermal properties of the formulations. These formulations were then applied onto the calorie cell model using the TSI to evaluate the release rates of the drug from the system and was compared to the France cell. With the France diffusion cell, dry powder formulations were deposited onto the filter membrane and microparticulates are rapidly wetted followed by diffusion into the surrounding media. Analysis of the spray-dried ciprofloxacin release from the France diffusion cell showed a rapid release profile with more than 95% of drug being released within the first hour, in contrast to the controlled-release ciprofloxacin where approximately 88% of the drug being released over a 4-hour period, which is what was expected. Interestingly, the release profile of the same ciprofloxacin formulations were found to be the reverse in the calorie cell model, where controlled-release ciprofloxacin showed a higher percentage of drugs being transported across the cell epithelium compared to the spray-dried ciprofloxacin alone. This was also in agreement with previous in vivo studies. Further live cell imaging studies revealed that the PVA within the controlled release formulation was able to stabilize the amorphous form of the formulation upon deposition onto the thin layer of mucus, while the confocal imaging with ciprofloxacin autofluorescing blue showed that the PVA also forms a gel-like material 15 minutes after deposition onto the cell that enables it to cover more surface area and hence exposing more cells to the ciprofloxacin, accelerating drug transport to occur. Therefore, the deposition of dry powder aerosols onto the surface of air interface monolayer by the modified TSI produces a more representative model to study individual mucosa particle interaction in terms of dissolution permeability to that occurring in the lung. Subsequently, we have also advanced our systems by integrating our models into impactors with multiple stages such as Anderson cascade impactor and the next generation impactor so that we can incorporate the different relevant cell types into the different stages. To do this, we have utilized 3D printing technologies to modify the collection plate so that it could house multiple snap wells containing cells and place it in the stage of interest. It was established that the modified plate does not have any impact on the aerosol performance of the formulations. This setup not only provides a fundamental understanding of where the drugs go in the lung, but also the different mechanisms of drug dissolution and uptake, and to study the effects of particle size on cellular function. In this particular study, we evaluated the potential of the ACI integrated model to study if the cellular uptake of aerosols delivered from different devices equivalent. The study compared the transport of salbutamol sulfate aerosols produced from a commercial nebulizer system to a Ventolin pressurized metered-dose inhaler and a rotorcap DPI. It was hypothesized that salbutamol from PMDI and DPI would have similar characteristics and that will be significantly lower than the nebulized aqueous solution of salbutamol. This is because the salbutamol in Ventolin PMDI is predominantly crystalline particles suspended in HFA propellant, and after actuation, the dried solid crystal aerosol particles would present a similar behavior to the DPI formulation, where salbutamol is present in crystalline form. Interestingly, it was instead found that salbutamol aerosols produced from pressurized metered-dose inhaler had similar transport kinetics to nebulized salbutamol that was significantly higher compared to the dry powder formulation. The differences in the rates of drug transport is in part due to the different physicochemical properties of salbutamol particles upon aerosolization and deposition. It was found that salbutamol particles produced from the DPI have an average size of 3 microns with crystalline structure and rectangular shape. On the other hand, particles of salbutamol produced by the PMDI formulation were smaller, approximately 1 to 2 microns, and comprised of a mixture of smooth spherical and rough-shaped rectangular particles, suggesting that the particle produced after aerosolization are a mix of both amorphous and crystal particles, which could promote drug dissolution into the epithelial lining fluid, resulting in enhanced drug transport compared to the DPI formulation. Indeed, it was reported that the PMDI formulation showed a tendency towards a better response in terms of improvement in forced expiratory volume in 1 second or higher salbutamol plasma concentrations. On the other hand, the high rate of transport of nebulized salbutamol is attributed to the rapid diffusion of the drug into the epithelial lining fluid, followed by absorption across epithelium, bypassing the need for the rate-limiting dissolution process. Similarly, we have also developed integrated models of the nose. For nasal formulation, the regulatory authorities have mandated the use of a nasal expansion glass chamber add-on to measure nasal and lung deposition. Hence, we have redesigned this glass expansion chamber using 3D printing so that the chambers consist of 2 separate hemispheres, an upper and lower hemisphere, where 3 snapwell inserts are incorporated into the upper hemisphere directly opposite the inlet hole to maximize drug deposition. Subsequently, this modified expansion chamber can then be adapted to fit onto the NGI as how the glass chamber did. Incorporation of the nasal cell model into the 3D printed nasal expansion chamber allows aerosols to be deposited on the epithelium in a physiologically relevant condition. Now, we have made several assumptions with the previous studies, but is there really a difference between the conventional approach, which is just pipetting drug solution and suspension on the apical chamber of the cells, compared to aerosol deposition approaches? To evaluate this, beclomethasone dipropionate was used to compare the transport rates of BDP using the conventional transport approach and the aerosol deposition approach. To take it one step further, we will also evaluate 2 BDP nasal formulations of the same dose, Rhinocort, which is a dry powder spray, and Beckinase, which is a nasal aqueous suspension spray. Besides wanting to compare the difference between the conventional and deposition approach by using different nasal formulation, we are also able to validate the expansion chamber for different nasal formulations and the effects of formulation on transport. In terms of validation, there is no significant difference in terms of mass deposited between the standard glass and modified expansion chamber. However, Beckinase nasal spray has significantly higher deposition of BTP in the nasal region compared to the Rhinocort dry powder spray. There is a difference after all. The conventional suspension approach showed the highest transport rate of BDP, which is most likely due to the dilution of the mucus layer on the epithelial cells, resulting in the loss of mucus barrier and hence the higher rate of permeation. As for the deposition approach, by the end of the 4 hours, it is approximately half the transport of the conventional approach, which is due to the non-homogeneous coverage of the deposition of aerosols on the epithelium. Although there was no significant difference between the 2 nasal formulations at the end of the 4-hour transport studies, but interestingly, the Beckinase nasal suspension had a higher transport trend of BDP initially in the first few hours compared to Rhinocort powder due to the saturation of some solubilized BDP in solution. Finally, how does the integrated model perform compared to other models, and more importantly, does it correlate to in vivo? A study comparing the release profiles of liposomal ciprofloxacin and free ciprofloxacin in 4 different methodologies were performed, including a dialysis bag, the cell-integrated model, isolated perfused lung, and in vivo mice model. Each of them have their advantages and disadvantages. The dialysis bag model was simple and straightforward, but the large volume of solution is not consistent with the airway physiology. The integrated air interface model is a simple and physiologically relevant biological system, but it does not possess the intact airway structure and full absorptive pathway that is present in the lung. On the other hand, the isolated perfused lung model maintains the structural integrity and functionality of the lung tissue, but has a very short viability period. And finally, the in vivo mice model, which continues to be the mainstay of assessment in pulmonary drug delivery, but has reduced screening capacity, marred by ethical consideration, and is expensive. So in general, similar trends were found across all the methodologies whereby the free ciprofloxacin have a higher transport rate compared to the liposomal formulation, but there were variations in the extent of transport. Mathematical modeling of the release trends showed that the free antibiotic formulation was diffusion driven, whereby transport was dependent on concentration and rate of transport decreases with time, while the liposomal formulation acts as a drug reservoir where drugs can be released at a rate limiting step. Consistent mechanisms of drug release and transport were recognized for the two formulations using the in vitro and ex vivo methods, and was also found to be predictive of profiles in vivo. Importantly, results from my model also correlated with clinical outcomes. The application and comparison of different methodologies are still required to elucidate the drug and formulation behavior at target sites, provide a complete picture of drug absorption, and also to establish a reliable in vitro and in vivo correlation. However, the simplicity, robustness, and experimental control available with the integrated cellular model provides a good platform for assessing the initial kinetic profile of controlled release formulations, and may be better suited than tissue models for identifying individual processes involved in complex and dynamic relationships across the different cell types. Therefore, the advanced integrated models developed so far have successfully identified fundamental factors influencing transport, toxicity, and efficacy, which has allowed my team to design new and repurposed inhaled therapies. It also reinforced that the potential of using in vitro cell culture model as a platform to elucidate the interactions and mechanisms to enable the development of safe and effective inhaled therapeutics. This includes new approaches for treatment of specific diseases, such as targeting lymphatic cells with nanoparticles, ensure prolonged residency time of antibiotics at the lung using liposomal formulation, combining different drugs together to reduce treatment burden, and using different smart excipients to enhance drug transport. Finally, I would like to share with you some of my current research that I'm working on in building the vision that I have for my future research. My research vision is to establish new tools and apply new methods to bridge basic science with clinical application to help drive respiratory science commercialization and translation to produce impactful outcomes for the benefit of healthcare sector and the community. One of the key focus to achieve this is to streamline research processes into integrated systems so that it is cost and time efficient. Concurrently, I will continue to develop new novel in vitro models as preclinical platforms with emphasis on real-life, physiologically relevant, and realistic models, while ensuring correlative results to in vivo studies. Ultimately, this will help accelerate translation of inhaled therapies that are safer and more effective to clinic. One of the current projects we are working on is to create a 3D physiologically relevant respiratory model that is anatomically accurate of the lung with capabilities to incorporate the different cell types to determine and visualize where the particle lands in the lung in a more accurate manner and study the biological effects of relevant inhalation doses. This will be performed using 3D designs and 3D printing technologies. In line with mimicking realistic environmental models, we have also developed capabilities into aerosolization of pathogen exposure such as Pseudomonas, dumbed-down coronaviruses, and Staphylococcus aureus to mimic airborne aerosol infection within a contained exposure chamber. This allows us to evaluate efficacies of strategies and measures to reduce spread of infection like mask or filtration devices. This setup is also applicable to other airborne hazards like urban dust and pollen exposure to mimic air pollution. Another area of active research is in microfluidic devices, where my team, in particular Hania and Janie, who presented at the Pat Brunel session, has created a reversible dual chamber design for culture of respiratory organ on a chip, as well as to study biofilms. These systems allow us to study the effects of flow conditions on cell growth, as well as their effect of different drug responses when cultured in different conditions that are more realistic to the real world in terms of interfaces, aerosol deposition, airflow, and pulsative flow. Our devices has also flexibility in changing the different interfaces and surfaces to mimic different environments, which is particularly relevant to study biofilm infections not only in the airways but also on medical devices, different environments like pipes, other organs, and plants. In terms of streamlining key research process, the microfluidic device has been incorporated with microsensors made using silver and carbon nanofiber mixtures to detect and quantify drugs. Currently, transport studies are performed over time where samples are taken at predetermined time points, processed, and then analyzed using HPLC, while TER measurements are performed using electrodes, which are endpoint studies that is performed at a particular time point, usually at the end of an experiment. These processes are time-consuming, requires lots of solvent, and may also disrupt the cell integrity. The microfluidic chip developed has capabilities to perform real-time monitoring of TER measurements over time using impedance and perform real-time quantification of drug transport using electrochemical technique. It was found that the electrochemical sensor method was able to perform as well as HPLC analysis. Finally, to help speed up translation of therapies to clinic, I have also co-founded and spin-off a pharmaceutical company, Ab Initio Pharma. It is a pharmaceutical formulation R&D and GMP manufacturing company based in Sydney, Australia, that provides innovators across Australia and globally with a flexible platform to rapidly translate benchtop discoveries to clinical trial product. The models established are an essential offering of Ab Initio Pharma services. Ab Initio specializes in taking a lead compound or idea to final product ready for clinical trial with a focus on inhalation products. Before I end, I would like to thank my team, the Respiratory Technology Group, for their ongoing commitment and support, and also particularly to my mentors, Professor Paul Young and Daniela Trainee, for their unwavering belief in my ideas and capabilities. Thank you for listening and happy to take any questions.